Our panel is going to focus on neurobiology and also changing paradigms in psychiatry. After me, our guest from Taiwan is going to talk about dopamine transporter and hippocampal volumes in schizophrenia. And Marek Kubicki is also going to talk about white matter pathology. And last but not least, Ali Saffet Gönül is going to be talking about an fMRI study about depression and emotions. These are my disclosures. These are the professors that we work together and also the financial resources. There are many reasons to work on schizophrenia and brain connectivity because in post-mortem studies, abnormalities in oligodendrocyte number and expression of oligodendrocyte markers and myelin-related genes and genetic association. When we look at the genetic association studies, oligodendrocyte function-related genes can be seen in schizophrenia. And last but not least, in vivo neuroimaging studies show us that especially in diffusion tensor imaging, DTI technique is being used in these studies, and all say that in the white matter, integrity is being disrupted. In the daily functions of white matter, there are some disruptions in schizophrenia, and of course, these disruptions change the communication of different parts of the brain, and the functionality of the brain changes as a result. And these studies have a long story. DTI technique has been used in these studies, and it has been a classical approach. And Kubicki, you see here, in schizophrenia, in many different locations of the brain, interesting disruptions can be seen in corpus callosum, for example, right up, and in the bridge, and also articulus fasciculus, AF, left up, and Wernicke's and Broca's bridging pathway, fornix, the exit pathway of hippocampus, and ancilis fasciculus, UF, medial temporal lobe is being connected to prefrontal cortex. And in schizophrenic patients, in these locations, there are disruptions in the white matter. DTI studies are based on the diffusion of water molecules. This was an important progress. The diffusion of water molecules is connected to the pathways of white matter. Therefore, the disruptions in white matter can be seen in diffusion water, but it's a non-specific marker because in the tissue, intracellularly and extracellularly, there is water, and also in the plasmic reticulum, there are freely diffusing water can be seen, the water within cytosol, and it has different characteristics, and of course MRI images are going to be different. We do have such an heterogeneity, and there is also exchange between compartments, and this mixes different diffusion properties. If we look at only water diffusion, it's not possible to understand where the problem is in the organ. And there are some other technical complexities of the approach. For this reason, when we look at the DTI literature up to now, yes, there is a disruption of water molecules in the white matter, but we don't know where this disruption is coming from the axon or from this red place or the white place you see on the right side of the screen. DTI alone can't tell us the biology of the white matter. In order to have more information in this regard, we started to ask questions. What else can we learn from diffusion MRI? This is scleroscopy It's similar to other MRI modalities. In this slide, you see the data of oscilloscopy, and on the left it's 4 Tesla, on the right it's 5 Tesla. There are peaks and valleys in between. All of these peaks is equal to one molecule. NAA is the biggest one. And the size of this peak is show us the NAA concentration in the brain. By using MRI stereoscopy, it's possible to get information about the brain. And diffusion technique can be used together with MRI, MRS. If we look at the diffusion tensor spectroscopy, NAA, it's an intracellular matter. Therefore, NAA is exclusively intracellular and almost totally in the cytosol, freely diffusive material. There is a small amount in mitochondria as well. Therefore, it's a marker of intracellular marker. In DTI, we can't see whether it's intracellular or extracellular, but with NAA, we can see the intracellular. And magnetization transfer ratio can be used. And with this technique, we can see how big the myelin compartment. We are looking at the NAA diffusion in the axons within the cells. There is myelin in the white matter, and in order to measure the amount of myelin, it's possible to use MTR. There are two different techniques here. DTI, the diffusion of water molecules. DTS is only looking at NAA diffusion within the axon, and MTR is measuring the myelin amount around the axon. When we combine them all, we can answer the question about the underlying reason. And this is the principle. You can see here NAA peak. The biggest peak is the NAA peak. And diffusion gradient during the MRI. NAA amount decreases. It happens just because of the diffusion. Apparent NAA amount decreases. The longer the peak, the bigger the amount of NAA. We compiled the studies we conducted in the last couple of years in the healthy axon diffusion, myelin normal, in the chronic bipolar disorder, in psychotic bipolar disorders. The patent is that axons are okay, NAA diffusion is okay, but myelin is decreased. In chronic schizophrenia patients, there is a disruption axon and myelin is decreased. Therefore, the pattern in the schizophrenia tells us that there is a disruption within the cell and out of the cell. And in bipolar, there is intracellular disruption. In schizophrenia, there are neural disruption, and also there is a decrease in myelin. But in bipolar disorder, neurons are relatively protected in spite of the fact that they are chronic, but myelin disruption is still there. Studies in first episode psychosis patients, we haven't published it yet. We will do it recently. The first attack is bipolar, not depression. These are manic patients. The first attack is schizophrenia, and we collected data from the controls. The age is quite young. You see them. Age onset 21, 90, 20. These are the first attack patients, and the findings are interesting, both in MTR and in NAA diffusion. We couldn't find any difference between the groups. We don't see what we see in chronic patients in the first attack patients, not in the NAA diffusion, but in creatine and chole diffusion we see disruptions. they exist in neurons and glia. NAA exists only in neurons. Neuronal marker is normal in the first attack, but the markers in both glia and neuron, they are disrupted in the first attack. Interesting pattern. And another correlation that attracted our attention is that in the controls, we couldn't find any relation between the age and colon diffusion, but in the schizophrenic patients, we have seen a correlation between the diffusion and in the colon disruption becomes normal in the patients with late attack, with the first attack at later ages. Conclusions: both axonal and myelin-related abnormalities in chronic stable schizophrenia, and only myelin reduction in chronic stable bipolar disorder. But there is evidence for non-neuronal metabolite diffusion abnormalities in first episode psychosis. But in terms of myelin content, it's normal, and axon geometry is also normal. Neurons are relatively protected, and myelin has not decreased yet in the first episode psychosis, but there's disruption in the glial abnormalities. What do we mean by saying them? In the first attack episode, it shows us that the processes, non-neuronal processes, are disrupted. It's not easy to make a comment on that, but when we say non-neuronal processes, neuroinflammation comes to your mind, especially in the first episodes of psychotic diseases. There are serious studies conducted about the infectious disease, and our findings are supporting them. And neurons in the first episode are protected relatively, but there's disruption in glia, maybe because of inflammation, maybe because of some other changes, because glia have many functions. It's hard to tell, but when it becomes chronic, glial processes are normal, but there's disruption in the neurons. In the bipolar, we see the disruption in myelins. It's an interesting process, an active process. White matter is very important to understand these processes in a better fashion. Thank you very much.